Hello, this is the continuation of the PowerPoint narration for cancer and hereditary disease, and this is, of course, the hereditary disease portion. And I want to remind you that in this course, and in any course, we cannot possibly cover all of the aspects of disease, of cancer, of genetics, of hereditary disease. So I am trying to help you focus upon what I feel is the important take-home, let's say take-home message for you. So we are really just skimming the surface. I want you to make sure that you understand basic definitions, and then we can proceed to having just some basis of understanding of the disease process and of various types of diseases, and in this case, a little bit about genetics. DNA, or deoxyribonucleic acid, is the genetic blueprint for our bodies, for any living creature. And there has been much work done on genetics, and now we do have a complete map of the human genome, which means we know what each chromosome contains and what information each chromosome codes for. There are 23 pairs of chromosomes in the human body, or 46 chromosomes. Contained on the chromosomes is the actual information, and each bit of information is referred to as a gene. Genes exist in pairs, and the pairs of genes that code for the same information are called alleles. 44 of our chromosomes are called autosomes, which means body chromosomes, and then 2 of our chromosomes are the sex chromosomes, the X chromosome, which are contained in the female as 2 X chromosomes, and the Y chromosome, which are contained in the male as 1 X chromosome and 1 Y chromosome. Y-bearing sperm will produce a male. X-bearing sperm will produce a female. All of your genetic information is referred to as your karyotype, or your genetic type. Alleles exist as either homozygous, which means coding for the same information, which means, for example, although it's a little bit more complicated than this, that for the color eye, blue colors in eyes, you would have both genes would code for the color blue, so they would be homozygous. If you were heterozygous, that means you would contain on one of your alleles slightly different information, perhaps a slightly defective gene. For example, in sickle cell trait, a person who carries the genetics but doesn't have sickle cell disease per se would be heterozygous. One of their genes for producing red blood cells would contain the defective sickle cell gene. The other would contain normal red blood cell producing genes, and this individual would be called, for this trait, heterozygous. If one of the alleles is expressed in the phenotype, for example, if you contain coded information for blue eye and brown eye, you will produce brown eyes, because the color brown, the pigment, is dominant to blue. Excuse me. So again, it's a little bit more complicated than that, but that gives you an idea. The gene that is not expressed, for example, in this example, the blue eye color, would be called the recessive gene. So this is kind of genetics 101 in about 3 minutes. This is basically what the human genome, or the human karyotype, looks like. What your genes look like as they are contained on your chromosomes. So these long bodies are the chromosomes, are the pairs of chromosomes. and the dark spots you see may be the location of the genes. All right, so in autosomal dominant characteristics, you would be transmitting a dominant allele, and each offspring would have a 50% chance of being affected by whatever disease condition. The disease would appear in every generation because you might have inherited the dominant gene, and in this case, the dominant gene does produce the disease. In this case, males and females would be equally likely to inherit the disease. Some examples of this are polydactyly, where you have extra digits, achondroplasia, which affects the cartilage, Marfan syndrome, which produces gigantism and some other serious conditions like an underdeveloped heart for the size of the individual, some facial characteristics, and familial hypercholesterolemia, which means an inherited tendency for very high cholesterol in the blood. This diagram shows you how an autosomal dominant trait is transmitted. So a person who is heterozygous carries one, let's say, recessive gene and one dominant gene will have the disease. This is a dominant trait. So if you have either allele that has this disease characteristic, you will have the disease characteristic. When sperm or eggs are produced, half of the sperm, in this case it is the sperm because the male has the disease, will carry the affected diseased, let's say, gene, or the gene that codes for the disease. In this case, they're showing a female parent who is unaffected. Notice how, because half of the male's sperm contains the, let's say, damaged gene, that it is possible that half of the children will be affected. And this is a diagram of how that works. Again, I'm not going to belabor this and won't test you precisely on genetics or making diagrams like this, but it is extremely important that you understand how these diseases can be inherited and what the difference is between dominant and recessive genes and diseases. An autosomal recessive, you only have the disease when you have both of the defective alleles. In this case, the parents would be carriers. They don't have the disease, but they would carry the disease. And a child would have about a 25% chance of being affected. The recessive allele appears more frequently in close intermarriages because you are likely, the closer you are genetically to your mate with whom you produce children, the more likely you are to carry these recessive traits that will be passed on. Here are some examples of the diseases that are inherited via this method of recessive autosomal diseases. For example, Tay-Sachs disease, which is a neurological disease and generally causes death within the first 2 years of a child's life, is predominantly found among Eastern European Jewish people who tended to marry very close in their family, perhaps cousins, for example. Sickle cell anemia is found in many people of African origin because having just the trait, not the disease, but just one recessive gene, would give some degree of resistance to malaria. And there's more about this in your book and in some of the articles I've included for you to read today, or to read this week. Sickle cell anemia is one of the diseases upon which we will be concentrating. Here is a chart which shows you how these diseases could possibly be inherited. The only affected children are those who have inherited both of the defective genes. So in this case, both parents would have to be carriers in order for, on the average of 25% of their offspring to be affected. There is a film on the Moodle page that you can click into that shows you some more information about sickle cell anemia. Sex-linked inheritance means the defective gene is on the X chromosome. The defective X on the male is unmasked and the trait is expressed because the Y chromosome does not contain any information for these particular diseases, and therefore we could say does not provide genetic dominance or genetic protection for this condition. So if you think about the Y chromosome as sort of missing this bit, that extra little leg of the X, missing this bit of information, then there is no dominant trait. If a recessive defective gene is inherited and nothing for that trait on the Y chromosome, the individual will have the disease. There are diseases also of abnormal chromosomes, altered numbers or structure, failure of the chromosomes to separate during cell division. Familial diseases run in families, and most likely the effects of several genes working together. Diabetes, allergies, familial, I should say, polyposis. Down syndrome is another disease that is caused by abnormal, let's say, genetic makeup. This is caused by the presence of an extra autosome. It's sometimes also called trisomy 21, which means 3 number 21 chromosomes rather than the normal 2. This tells you some of the characteristics of that condition. Often, females who are older will pass along this condition because during the production of their gametes, the 2 number 21 chromosomes do not separate, and therefore when the sperm carries the number 23 chromosome, the offspring will have 23 of those chromosomes. How do you diagnose genetic diseases? Here are some ways to do that: amniocentesis or chorionic villus sampling. Congenital diseases appear at birth. Congenital means a disease that appears at birth. They do not get passed on generally to offspring because they are a failure in development rather than a failure or an abnormality of genetics. Here are some examples of the conditions that are inherited as congenital diseases: spina bifida, cleft lip and cleft palate, and pyloric stenosis, which means the pyloric valve in the GI tract is not open. All right, so on Moodle there are films on the following topics for your review: the genetics of cancer, leukemia, breast exam, breast cancer, mammography, sickle cell anemia, and Down syndrome, which is also called trisomy 21. There is a lot of information. We're just skimming the surface. Please feel free to send to me any questions you may have, and lots of success to you.